revised the topics that we have discussed yesterday in the class, right? So in yesterday's class we have covered the human circulatory system. We have read about the blood, the formed elements and the plasma, that is the composition of the blood, and apart from that we also have studied the functions of the circulatory system. Right. So, and we also discussed in the formed elements we mainly have three types. Okay, we have this RBC, we have this WBCs, and then we have the platelets. Right. So, in the WBCs we have again the granulocytes and the granulocytes, and again we have discussed the subtypes of it. Okay, so quickly let us recall. So what all come into the category of these granulocytes? We have the basophils, we have the eosinophils, and then we have what? The neutrophils. Now what comes under the category of the granulocytes? That is the monocytes and the lymphocyte. Okay, so apart from that we have even discussed about the platelets. So what exactly is the role of the platelets? Now here the platelets are also called the thrombocytes. Now they mainly help in the clotting of the blood, right? So on this yesterday's topic we have few questions. Now let us quickly solve these questions. Let us move on to the first question. Now which of the following, now let us read out the first question. Which of the following organisms has an open type? The open type of circulatory system. So what exactly happens in the open type? So yesterday we have discussed in open type of circulatory system we don't have the well developed blood vessels, and the blood directly flows into the cavity, the body cavity called the hemocoel. And what is the fluid called? The fluid which is flowing into it is called the hemolymph. Now in which it is observed mainly in what? The arthropod and the mollusk. Okay, so here what exactly comes under the category of the arthropod? See, the earthworm is an annelid, right? So the cockroach is what? It's the arthropod. So that would be the answer. Whereas the human, okay, it comes under the phylum, the chordata. Okay, now whereas the frog, okay, it's also the chordate, it belongs to what? The amphibia. Whereas the earthworm belongs to the annelida. So here the correct answer would be the second option, that is what? The cockroach. Now let us quickly move on to the next question. Yeah. Now in a closed circulatory system the blood flows through. So here in the closed circulatory system the blood flows through. The blood flows through what? The blood vessels, right? And it forms a closed loop connection over there. So what's the answer? The tissues directly. Now this happens in what? In the open. Next, the heart only? No, it's the blood vessels. And is the lymphatic one? No. Now what flows into the lymphatic vessels? It is the lymph fluid. So in today's class we'll be discussing about the lymphatic system in detail. Now next, which of the following is not a component of blood? Now yesterday we have studied the blood is made up of what? The plasma and the formed elements, right? So here what exactly are they? Let us see. The plasma, yes. Okay, is lymph? No, lymph is not the composition of it. Okay, so it would not be correct. Then we have the plasma and then we have the WBC. Okay, so they are asking what is not. So here in this case the answer is what? The lymph. Next, let us quickly move on to the next question. Now here they are asking the iron containing pigment which is present in the RBCs. What is an iron containing pigment which is present in the RBC called? Okay, it is called myoglobin. No, it is present somewhere in the tissues. It is hemocyanin. No, it is present in what? It is present mainly in the mollusk and all, but not here in the RBCs. Okay, so we have the hemoglobin. The hemoglobin, right? And also hemoglobin is also incorrect. Let us quickly move on to the next question. Now which component of blood is mainly responsible for clotting? Now yesterday we have discussed about the clotting. Now whenever there is an external injury, right? So here to prevent the loss of blood from the body, what exactly happens? Okay, so here this process clotting happens. So here it will be preventing the blood to flow out or prevent the blood loss from the body. And the main the formed element which plays a very important role in this is the platelets, which is also called the thrombocyte. Fine. So the RBCs, no. WBCs, no. The platelets, yes. And the plasma. So the platelets are nothing but they are also called the thrombocytes. Fine. Now let us move on to the next question. Now which blood cells help in defense against the infections, like to improve the body's immune responses? Okay, which one it will be? So here, is it the RBCs? No. Is it the platelets? No. It's the WBCs. Okay, so again in the WBCs, if they specifically ask you which part, it would be the lymphocytes. Okay, because the B lymphocytes, they help in the production of the antibodies, and the T lymphocytes, they help in generating the responses to against it, and it will not be the all of the above. Next, now the red blood cells are also known as, okay, so what are the red blood cells also known as? The red blood cells are also known as what? The erythrocytes. Fine. Okay. Next, it was an easy question. Now here they are asking what is the lifespan. Okay, so what is the lifespan of the red blood cells in the human body? Okay, approximately. So the lifespan, the average lifespan of the red blood cells in the human body is 120 days. Okay, it is how much? 120 days. So here which option? 60 days? No. 120 days? It's not the 30 days, it is not the 10 days. Let us move on to the next question, the hemoglobin. Now hemoglobin is a pigment, right? Now where exactly this hemoglobin pigment present? Okay, so hemoglobin is present in plasma. No, it is not present in the plasma. It is present in the red blood cells. Okay, now here it is not the WBC and not the platelets. Next, moving on to the last question, the blood plasma is mainly composed of. So yesterday we have discussed, right? Now what exactly the plasma is made up of? The plasma is made up of what? Okay, we have it constitute of the water. Okay, it is having the proteins. Okay, how much of water? We have the 90 to 92% of the water, 6 to 8% of the proteins, and the minerals are present. Okay, so here the cells and tissues, no. Water and the proteins, yes. Enzymes only, no. The DNA and the RNA, no. Now what are the proteins which are mainly present? We mainly have 3 proteins, right? What exactly are there? So the 3 proteins which are present, we have this albumin, we have this globulin, okay, and then we have this fibrinogen. Okay, we have this fibrinogen. Fine. So now today let us start the new topic, that is the ABO blood grouping. Fine. So students, I think all of you, okay, at any point of a time had gone for the blood test. Okay, and even you all know your blood groups, right? So here how exactly we can tell what exactly is our blood group, what makes it different from the other blood group. Okay, and who was the scientist who discovered for the very first time about the blood group? Now let us discuss all these topics quickly in today's class. Fine. So here let us start the new topic, that is the ABO blood grouping. The ABO blood grouping. Now who was the scientist for the very first time discovered about the ABO blood grouping? It was the Karl Landsteiner. Okay. It was the Karl Landsteiner who for the very first time discovered that we have different blood groups. Now have you ever wondered how he got to know about this different type of blood groups? So during their situation, now whenever there was an external, like whenever there was an injury, so here what happened, now the man suffered with an injury, and due to that injury there was a heavy blood loss. Okay, so he as a doctor, so what exactly he did, he made the blood transfusion. Okay, so the one without checking any blood group, randomly the blood was what? Okay, it was transfused into the patient's body. Now slowly what happened? Okay, so slowly they observed that the clumping, they started observing the clumping in the blood. Okay, so whatever blood that was transfused, it slowly started clumping in the body, and soon what happened? Okay, the patient condition got little complicated, critical. So here the scientists got to know that we have the different kind of blood groups, okay, which and we need to transfuse it accordingly. Now if there is not compatibility between the blood groups, so in this case what will happen? So in this case the clumping of the blood happens in the body. Okay, so we have how many different types of blood grouping? We have the A, we have the B, we have the AB, and then we have the O blood group. Yes, so how many types of blood groups do we have? Okay, so we have this A. Yeah. So we have mainly 4 types. Mainly 4 types of blood group. What are they, students? So we have the blood group A, the blood group B, the blood group AB, the blood group O. Okay, so we have mainly 4 different types of blood group, right? Now here you may all wonder that ma'am, how exactly we can know that only the A person, the A blood group can donate it to the other, okay, to this A itself and also the AB. What is the factor, right? What exactly makes the difference? Now how this process exactly will be happening? Now let us have a quick overview of this. Fine. Now how exactly this process happens? Now before getting to know about this, so see students, what exactly is an RBC? Now RBC is a cell. Okay, so we have this RBC. What is RBC? The red blood cells or the erythrocytes, right? It is a cell in itself. Means what are the components of the cell? The cell is having what? The cell is having the plasma membrane. It is having the mitochondria, all the cell organelles which are present, right? In the similar way, now here on the cell surface of the RBC, okay, so what exactly is present? We have this antigen. Okay, so we have the antigens which are present on the RBC. Okay, so what exactly is an antigen? Fine. So I think you are very new to this word. We have the 2 terminology. We are using the 2 words, antigen and the antibody. Antibody. Fine. Now for example, see, whenever there is a donation, okay, whenever there is any kind of a transplantation, okay, so for example, just imagine there is a patient who has undergone the kidney transplantation. Now from whom they will be taking up this the kidney, on what basis? So the one donor, they need to match, right? The graft matching need to be there. Right. In the similar way, when we are saying the blood transfusion, okay, even in this blood transfusion also what will happen? Even in this blood transfusion also, the particular, the one who are suitable or the compatible only can donate. Now what exactly is an antigen? Now antigen is something which is present on the cell surface. Okay, so we are multicellular organisms. So in our body we have many different type of cells, right? So on each cell we have the antigen. Right, so this specifies that this particular cell belongs to our body. Okay, so here it helps our immune system to detect whenever a foreign cell, or a foreign pathogen enters into our body. The foreign pathogen is having a different antigen. So what our body's immune system will do? It will be detecting the foreign antigen which is present on the particular pathogen, and it will be passing on the signal. It will be passing on the signals to what? The body's B lymphocytes, okay, to produce the antibody against it. So here these antigens, it will help our body, okay, help the immune system to distinguish, okay, the self and the non-self cells, or you can say the self or the foreign cells which are present in our body. Got my point? Yeah, so what exactly is an antigen? Now these are present on the cell surfaces, right? Now which will help the immune system to, you know, identify that this exactly is a cell belongs to our body. Now what exactly is an antibody? Antibodies are the proteins, okay, these are produced by what? These are produced by the plasma cells of B lymphocytes, and they are produced against the antibody. Okay, so whenever an antigen comes in contact with the antibody, for example, this is an antigen, right? And here this is an antibody. Now whenever the antigen comes in contact with the antibody, whenever they come in contact with the antibody, what will happen? Now it results in clumping. It results in clumping. It results in clumping there. Fine. So what exactly is, do we mean by this clumping? What exactly do we mean by this clumping? For example, now this is a normal cell. This is a normal cell. So whenever there is a clumping, so what will happen? Now here this will be open, and all this will be, you know, open, and all the cell organ and all the organelles, the components of the cell will be which will be like released out from the cell. Now this process is called what? The clumping, or you can say the lysis of cell. The lysis of cell, or you can say what? The clumping. Fine. Okay. So here this is the clumping. Now here after that, few of them even they would be stick to each other. Right, all the cells will be sticking to each other, which later on causes what? The lysis. Okay, so this is what you call the clumping there. Now let us understand how exactly this process happens. So here I think you are now clear with the terminology, what exactly is an antigen, okay, and what exactly is an antibody. So antigens are present on the cell surfaces. Now what exactly are the antigens? You can write the antigen, and we have the antibody. We have the antibody. Now what are antigens, students? Now antigens are present. They are present on the cell surface and helps body's immune system to distinguish self cells, okay, the body self cells and foreign cells, and the body's foreign cells. Okay, so whatever that is being entered, that is a non-self for the foreign cells. Now what exactly is an antibody? What exactly is an antibody? Now here, okay, so it is produced by plasma cells of B lymphocyte, okay, plasma cells of B lymphocyte against the antigens, okay, against the antigens present or entered into the body. Now what happens now during the antigen and antibody reaction? Okay, so what happens during the antigen antibody reaction? Now what happens when an antigen comes in contact with the antibody? What my point? Now for example, this is the antigen and this is an antibody. Fine. Now where exactly they are present? These are present, antigens are present on the cell surface. I am telling you like repeatedly that antigens are what? They are present on the cell surface. Now these are what? The antibodies. Where are the antibodies present? produced? They are produced by the plasma cells of B lymphocyte, and where they are present? They are also present in the plasma. Okay, you can write down, they are present in the plasma. Okay, they are present what? They are present in the plasma. Now what happens in the antigen antibody reaction? Okay, so when an antigen reacts or binds with the antibody, it results in clumping. It results in clumping there. Okay, so how exactly it will happen now? For example, this is the antigen which is present on the cell. Okay, now this is the antibody which is coming in contact. Now what will happen to this cell? What will happen to this cell? The cells will undergo clumping. The cells will undergo clumping. Okay, fine, which is followed by what? The lysis. Fine, I hope this part is clear. Now let us understand what antigens and what antibodies are present on the different blood groups and how it will be a compatible match. Okay, so let us have a look quickly. Now let us read about, so let us quickly draw the table. Fine, so here, yeah, so here what we are writing? So here we are writing the blood group. Okay, the blood group. Next, the antigen, and then next the antibody. Fine, and then let us write down the donor. Okay, to whom it can donate. To whom it can donate. Fine. So here the donors and who would be the recipient. Let us just do it even parallelly. Now here what is the first blood group? Okay, so let us start with the first one, that is the A blood group. We have the B blood group. We have the AB blood group, and then we have the O blood group. Now next, what did I tell you, students? Where do we have these antigens? Where these antigens are present? Okay, they are present on the cells of all types, but here in the ABO blood grouping, we are taking which cell? We are taking the RBC. Okay, so this is what? This is your RBC. Fine, this is what? Your RBC. Okay, so I think we missed out O. Yeah, so these are your RBCs, students. Okay, now next we are taking the antigen. Now let us take one color for each antigen. Fine. Now for example, what are the antigens which are present on the A blood group? Okay, so here they have an antigen, that is the antigen A. Okay, what is the antigen? The antigen A. So this is the antigen which is present on the cell surface, and this is antigen A. Clear? Now what is an antigen which is present in the B blood group? Okay, what is an antigen which is present in the B blood group? We have the antigen B. We have what? The antigen B. Okay, next, what are the antigens which is present in the A blood group? AB blood group. Okay, so that is what? Both the antigen A and antigen B. This is what? Your antigen B, and this is your antigen A. Okay, the antigen A. Fine. Now, what are the antigens present here? No antigens are present. And I'll tell you a fact. Okay, I'll tell you a fact. You know that why it is called the blood group A? Okay, why it is called the blood group B? Why it is called the blood group AB? Why it is called the blood group O? Okay, why it is called the blood group O? Fine. So, do you know, students, here because of the presence of the antigen A on the cell surface, it is called the A blood group. Okay. So, on what basis we are categorizing or we are classifying the blood groups? Now on the presence of the antigen on the cell surface. So here you can see if the antigen A is present. Okay, if the antigen A is present on the cell surface, then that is what? That is the blood group A. If antigen B is present on the cell surface, that is called what? The antigen B. If the antigen A and antigen B both are present, that is called which blood group? That is AB blood group. If no antigens are present, then what? If here no antigens are present, okay, no antigens are present in the cell surface, then what it is? No antigens on cell surface. Okay, no antigens on cell surface. So that is which blood group? Okay, that is O blood group. Okay, so this is clear. See, I am explaining you one more time because this is a very fundamental concept. Okay, if you understood this part, then we will move on to the antibody one. Now, why it is called the blood group A? It is called the blood group A because of the presence of antigen A. Why it is called blood group B? Because of the presence of the antigen B on the cell surface. Why it is called the blood group AB? Because of the presence of what? The antigen, both the antigens, the A and the B. Okay, and why it is called the O blood group? Because no antigen is present on the cell surface. Fine. Now what did I tell you? Antigen is present on the cell surface, and where the antibody is present. Now you can write down antigens present on cell surface. Where are the antibodies present? The antibodies, they are present in plasma. Where they are present? In plasma. Got my point? Okay, where they are present? They are present in the plasma. Now yesterday I told you what exactly is the plasma. The plasma is the fluid medium. Okay, the fluid component of the blood is called the plasma. Fine, it is called what? The plasma. Now let us here consider this is a plasma. Okay, this is your plasma, students. Let us consider. Okay, now this is your plasma. Now just before telling you what antibodies are present in the plasma, I'll tell you one thing. Now let us quickly go little back and revise. What did I tell you? I tell you, for example, if antigen A comes in contact with antibody A, so what would happen? There would be a clumping. Okay, so in our body, if we have the antigen A on our cells, so will there be an antibody A in our plasma? Just think logically. So if the antibodies A is there in our plasma, then at that time what will happen? It will be resulting in the clumping, right? So always the antagonist, that is the opposite antibody, is present in the plasma there. For example, let me tell you for the first one. Now here we have antigen A. So what antibody is present? Okay. Now what antibody is present? The antibody is antibody B. In the A blood group, students, I am telling, in the A blood group, on the surface, if you have antigen A, in the plasma you have antibody B. You don't have antibody A. If you have antibody A, at that time what will happen? At that time there would be a clumping. There would be clumping of blood. Getting my point? Now what is this? This is the antibody A. This is antibody. You have the B there. Fine. Now for example, in the B blood group, we have B antigen on the cell surface. What antibody need to be in the plasma? Okay, so if we have the B antigen on the cell surface, okay, so what antibody we need to have? The opposite one. That is which one? The antibody A. Fine. Now why we have the different antibodies? Just to prevent the clumping. Okay, getting my point? Just to prevent the clumping. Now here we have what? We have this antibody A. Now you may have a doubt that why am I drawing this structure? Now to specify this, I will tell you this is structure of the antibody, and you will be reading about the structure of the antibody in the immunity chapter that we will be discussing in the upcoming days, okay, the upcoming classes. So next we have what? we have the two antigens. Now just see, we have two antigens, antigen A and antigen B. Now just imagine if they have both the antibodies, that is antibody A and antibody B, will they be a clumping? Okay, will they be a clumping? For sure, right? Even you can't take any one. Okay, even in that case they would be a clumping. So what exactly is there? So here in this AB blood group there are no antibodies. Okay, so they are what? We have no antibodies. There are no antibodies present in the plasma. This is clear? No antibodies in the plasma. Next, we have no antigens on the cell surface in the O blood group. When we have no antibodies in the cell surface in the O blood group, then they are both the antibodies present in what? Present in the plasma. Getting my point? So here I can do the two of the antibodies. What are the two antibodies? Okay, the two antibodies are your antibody A and antibody B. Okay, this is what your antibody A. Okay, and this is what your antibody B. Antibody B. Fine. So understood, students? So did you understood? Fine. So what antigen is present on the A blood group? In the A blood group, the antigen A is present on the cell surface, antibody B in the plasma. In blood group B, we have antigen B on the cell surface and we have antibody A in the plasma. In the blood group AB, we have antigen A and antigen B both on the cell surface and we have no antibodies in the plasma. Okay. In the blood group O, you have no antigens on the cell surface. Okay, what you have two antibodies in the plasma. Getting my point? Fine. Okay, so let us quickly write them down in the tabular form. Okay. Yes, so here what we have, the blood group, next the antigen and the antibody. So here the blood group A, what antigen is present? A antigen. What antibody is present? Antibody B. What is the blood group? Blood group B, what antigen is present? The antigen B, the antibody A. What antigens are present in the AB blood group? Both the antigen A and B. What antibodies are present? No antibodies. No antibodies. Next, what is the O? In the O we have no antigen. We have no antigens, but we have both. We have both A and B antibody. We have both A and B antibody. Getting my point? Fine. Now this is clear. Now on the basis of this, let us understand the blood grouping. Fine, let us understand the blood grouping. Now just imagine, students, please have a look here. Okay, let us quickly have a look here, right, and just tell me that which blood group can be donating. Now for example, you have the blood group A. Now the blood group A is having what? The blood group A is having the antigen A and the antibody B. Now tell me for example, what are the other blood groups who can receive the blood from A? So A can donate to which blood group? For example, if A is donating to A, any problem would be there? No problem, na? The A blood group person can donate the blood group to the, the person with the same blood group, right? Because see, the same antigen and the antibodies are there, that would be no harm. Next, can B blood group, can A blood group donate the blood to B? Okay, this is possible, right? Now, can A blood group donate it to B? Now, just see what is present on the cell surface of antigen. The antigen A is present. Now, what is an antibody present here? Okay, the antibody A is present. Now, just imagine if the blood group A, which is containing, so here the RBC is containing the A antigen, they enter into the plasma. What will happen? So the antigen and antibody would react and it would result in what? It would result in the clumping of the blood. Isn't it? It would be resulting in what? It would be resulting in the clumping of the blood. So is it advised for receiving or donating the blood? No, right? So here the A cannot donate the blood to B. What about the AB? Okay, what about the AB? Now see, the A, what is the antigen present? Okay, the antigen present is A. What is an antibody? The antibody present is B there. Okay, but can it donate? Now what will happen? Now for example, if the antigen A is going into what? The AB blood into the body. What is present in the plasma? Do they have any plasma? Do they have any antibodies in the plasma? No antibodies in the plasma. So if there is no antibody, there is nothing to react with the antigen. So there will be no reaction. Right? There will be what? The no reaction. So in this case, what will happen? In this case, the A blood group can donate the blood to what? The AB. So without any clumping. Okay, so here there is a compatibility. So yes, this is possible. What about the O? Can it donate to O? What exactly is there in the O blood group, students? Okay, so here they don't have any surface antigens, but they have both the antibodies in the plasma. Right, they have both the antibodies in the plasma. Now for example, if the A blood group is getting transfused to O, so these antigen, the RBC containing the antigen A will go into the plasma. Now what is present in the plasma? The antibody A and antibody B. Now what is the antigen present here? Antigen A. So here when they go into the plasma, what will happen? There is a clumping. The antigen A and the antibody A would react and form the clumping. So here also it is not advised. Here also it is what? Not advised. So what we can say? The A blood group can donate the blood to what? The A blood group can donate. Who would be the donor? It would be the A itself along the AB. Is it clear? Yeah, it can donate to itself, okay, and apart from that the AB there. Fine. So this is the donor. Next, I hope this is clear. Let us go for the next blood group. The next blood group is the blood group B. What is present on the blood group B? Which antigen is present in the blood group B? Tell me which antigen is present. Now the antigen present in the blood group, blood group B is the B antigen. Okay. Now here we have the B antigen. Now can B donate to A? Can the B blood group donate the blood to A here? Now what is present on the cell surface? The antigen B. Now where it will be going? Now here if it goes into what? The A. So what is present? Here we have antigen B. Now if it goes into the A, so B blood group person, if he donates the blood to A, okay, the person with the blood group A, so what will happen? Now here these antigen B will be going into the plasma of the person with the blood group A, but what is present in the plasma? Antibody B. So there it will be a clumping. Okay, so in last what would happen? It will not be efficient. So here this is not a good match. Fine. So here it is no. So the B cannot donate the blood group to A. Can B donate the blood group to B itself? Yes, antigen B, antibody A. So see, it should be always opposite. If it is same antigen and same antibody, there would be a clumping. If there is an opposite antibody, that is antigen A, but the antibody is B, then there would be no clumping. So in this case, the person can go ahead and donate the blood. Yes, it is possible. Can B donate to AB? Yeah, AB is what happen if it goes to, yeah, this is not suggested. This is suggested. What happens? What will happen if they will be AB, it goes to the AB. So in AB, what is the thing? So in AB, they don't have any antibodies. Fine. So for example, even if they donate also, what would happen? Okay, as there are no antibodies, so there would be no clumping. So even in the case of AB, they can go. But what about the O? In the O, what happens? In the O, on the cell surface, we have no antigens. But what we should look for? We should look for this, we should look for what? We should look for the plasma. What exactly is present in the plasma? We have the antibodies. Fine. But when it goes into the plasma, when it will be going into the plasma, what will happen? Okay. So here as they having antibody both A and B, so there would be a clumping there. So that's the reason it even cannot donate to O. Okay, it cannot be donating to O. Fine. So what are the blood groups at the last? The blood groups at the last is what? The B and the AB. Okay, this is clear. I hope you have understood how exactly we can check the compatibility, right? Okay. Now next, moving on to the next blood group. What is the next blood group? The next blood group is blood group AB. Why it is called the blood group AB? It is called the blood group AB because of the presence of two antigens, that is both antigen A and antigen B. Now just let us see, can AB donate the blood to A? Fine. Now see the structure of, Okay, so now when it goes into the plasma, what will happen? Now they have an antigen B, the plasma you have antibody. So can it donate? What happens if it donates? Okay, so there would be again a clumping. So this is not a good match. Okay, so this is something no. So they can't donate. Next, what about the blood group B? Can it, so can it donate to blood group B? So what is present here in the B, you see, right? So here when they donate it to the B here, so they have the antibody A and they have an antigen A. Again there would be a clumping. So again here it's not a match. Now can they donate to itself AB? Yes, because they have no antibodies and no antigen. So again there will be no clumping, so they can just go ahead. Now what about the O? The O is not having any antigen, but as they are going into the plasma, what will happen? So if this goes into the plasma, what will happen? Both antigen A and B are present on the A, okay, on the cell surface, but when it goes into the plasma, there are antibody A and antibody B. So again there would be a clumping. So here even they cannot donate. So what we can say, the AB cannot then donate blood to any blood group, but it can receive the blood. Fine. So that's the reason we are saying it's what, the universal recipient. Fine. Okay, so it can receive the blood, but it cannot donate the blood. So to whom it can, donate the blood there in this case? Okay, so here it can donate the blood, to only itself, and it is a recipient. So it can receive from what? It can receive from A, it can receive from B, it can receive from AB, and even it can what? Receive from the O. Getting my point? It can receive from A, it can receive from O, it can receive from AB and O itself. Fine. Okay, so that's the thing. Next, moving on to the O part. The O is not having any of the antigen on the cell surface, but the main thing is it is having what? The antibodies in the plasma. Fine. So that's the reason here, for example, if A comes, so for example, if A is donating the blood to O, they have an antibody, so again there would be a clumping. If B is there, again there would be a clumping. If AB is there, again it would be a clumping. So O can donate the blood only to itself. Fine. Okay. It could donate the blood to itself. Fine. Okay, but here that's the only thing. Fine. So here you can say that what exactly is in, who is the recipients here. Okay, so here the recipient, it can receive the blood. So here in the case of this, it can donate to A. It can donate to what? Your AB. In the similar way, it can what? The B and AB. Right, in the similar way here it can receive the blood. It's called the universal, the recipient, whereas O is called what? The universal donor, but at the end it can receive the blood by itself only. Got my point? Now let us move on to the next part. Yes. Okay. We have the recipients, we have the A, we have the B, we have the AB, we have the O. Here we have the A, we have the B, we have the AB, we have what? The O. Okay. Now this is the recipient. Now here A is a donor. A can donate to what? A can donate the blood to A. It cannot donate to B, it can donate to AB, it cannot donate to O. But what about B? B cannot donate to A, it can donate to B, it can donate to AB and not to O. What about the AB? Now AB here, it can only donate to AB. Right. What about the O? O can donate to A, because why can A donate? Because here they have no no antigens on the cell surface, right? That's the reason. Now here it can donate to AB, it can donate to O. But coming to the AB, it cannot be the A. Okay, B, it is only the AB and it is what? The O there. Fine. That's the reason the O is called what? The O is called the universal donor. It is called the universal donor. Fine. Right. And whereas the AB is called the universal recipient. Is the recipient. Fine. Now for example, if you are a person with a blood group AB, you can get the blood from any other blood group. Okay, all the blood groups like A, B, AB and O. But if you are a person with a blood group O, you can only take the blood from the O blood group. Okay, that's the reason the O blood group, okay, they are called the universal donors because they can donate it to all other blood groups, but only can receive from what? The O itself. Okay, next, whereas the AB is what? AB can receive the blood from the all blood groups. That's the reason they are called what? The universal recipient. Okay, so apart from that, you may also have a doubt, right? Ma'am, we have even listened about this RH typing. Okay, so my blood group, okay, you may tell like my blood group is, A positive, B positive or, B negative, AB negative, right? Now, ma'am, what makes it positive and negative? Fine. Now, let us quickly read about that. Now, that is a rhesus factor. What exactly it is? It is a rhesus factor. Okay, that is rhesus, right? The RH factor that comes from the word rhesus. Now here this type of is observed from what? In the ancestors. Normally it was spotted in the rhesus monkey, right? It was observed the same in what? The rhesus monkey. Now ma'am, what exactly is this RH? Right? The RH is also a type of an antigen which is present on the cell surface. Now what is this rhesus? Okay, this is also what? The antigen. Antigen present on the cell surface. Fine. Along with the normal antigen. It was found in what? The rhesus monkey. The rhesus monkey. Fine. Now for example, if now I'll tell you, I think you all are used to it now, but let me just quickly draw. So I'm using here the color red for drawing the RBCs. Okay. Now here students, this is what your the A blood group. This is your B blood group. Okay. So this is your AB blood group and you don't have anything there, right? Okay, so what are these? These are your RBC. Okay, these are all your RBCs. So now this is what? This is your blood group A. Now what is the blood group? Yeah, so let me write down the blood groups here. The blood group. Now the blood group is A. The blood group is B. The blood group is AB. The blood group is O. This is clear? Yeah, this is clear. Now, for example, I am telling that I am taking one more antigen. What is the name of this antigen? Now, this is the RH factor. This is the RH factor, that is an antigen. What it is? It is an antigen. Okay, it is an antigen. Now, for example, if this is present along with the A, if RH is present, then you make it as A positive. If here it is present, this is B positive. Here it is present, it is AB positive. Here if it is present, it is O positive. Getting my point? Yeah? Okay, this is clear. Now, let me just do it. Yeah. Now, for example, we have this. Okay. Now, this is your antigen. This is your antigen B. Okay, so let me write down here. This is your antigen A. This is your antigen B, and the one which we are using right now is what? The RH antigen. Okay, the RH antigen. Fine. Now, please observe this. These all are again your RBCs. RBCs, these all are the RBCs. Okay, now tell me, okay, so here, what is the blood group, students? This is what your A, right. Now, here, let me draw this RH, all RH here, yeah. Now, if the RH present, now see, if antigen is present, then that is what? Then that is positive. Now, if this antigen is absent, if it is absent, then that is what? Negative. Getting my point? So, wherever you see this antigen, mark it as positive. Wherever you don't see this, you mark it as what? The negative. Okay. Now, let us start from this point. Okay, let us start from here. Now, what is present? Antigen A is only present, right? The only antigen A is present. So, this is what? This is your A positive. What is this? The A positive. Got it? Yeah. So, this is your A positive, A negative, because you don't have this RH. This is the A positive because you have what? Because you have this RH factor. I'm talking about this. This is the B antigen. So, this is B negative. Why negative? Because RH is not present. This is what? The B positive. Next, you have this AB both. So, that is why you write AB negative. You don't have antigen. Here, you have this RH antigen. That's why you write it as AB positive. In the similar way, this is your O negative. Okay, whereas this is your O positive. Fine. So, you have this different type of blood groups. Now, again you have one more major question, right? Ma'am, can a A positive donate the blood to the A negative? And can A negative donate a blood to the A positive? See, what did I tell you? Whenever an antigen enters into the body, what happens? A foreign body. So, at that time it start producing the antibodies, right? This is a basic concept. Past all this, we have been discussing that only. For example, if RH negative is there, okay, so this is negative. Now, if a negative person is donating a blood to positive, is there any RH antigen present on his cell surface? No RH antigen is present. So, when there is nothing present on the cell surface and it is being donated, so at that time our body will not recognize. Our body will only recognize if there is a presence of what? If there is a presence of the antigen, right? So, here, yes, they can donate. So, I am telling you what? I am telling you that the RH negative can donate the blood to RH positive. But can we do it vice versa? Can we do it vice versa? For example, can an A positive donate to the A negative? Now, positive means what is present? Positive means RH antigen is present. Okay, now when it goes into the A negative, on A negative there is no RH antigen. So, at the time what will happen? The body starts making antibodies against it and it will cause the lysis of the cell. So, in this case it is not possible. So, the A negative can donate, but the A positive cannot donate to what? The A negative. Getting my point? Yeah. So, I hope this is clear. Let me just write it down. So, there is a first question. What is the first question? Can the A positive, can A negative donate blood to A positive? Next, can A positive donate blood to A negative? So what would be the answer? Okay, so here the negative, okay, the A negative can donate the blood to what? Yes, you can donate because of absence of RH antigen on cell surface. Okay. What about this? Can positive donate to negative? No, because of the presence of what? Because of the presence of RH antigen. Okay, because of the presence of RH antigen, they cannot donate. Now, if they donate, what would happen? If they donate, it would result into what? The clumping. Fine. Now, because of the presence of RH antigen, which, okay, causes the clumping further. Okay students, so I hope this topic is crystal clear for all of you, right? And I'll be just quickly showing up the slides. Take the quick screenshots, right? Let us do it from the beginning. Okay, so this is the first that we have discussed, ABO blood grouping, okay, followed by, yeah, the structures. How exactly are the antigens and antibodies are present? The antigens are present on the cell surface, whereas the antibodies are present in the plasma. Next, it's a quick table. Next, yeah, so the recipient and the donor, and why the O is called the universal donor and why the AB is called the universal recipient. Next, we have this rhesus factor, we have this RH factor. Okay, so we have seen the blood grouping, right? And we have also seen the compatibility. Now, how each of them would look, okay? So, and we have also answered the question like why the RH negative, okay, can donate the blood to the RH positive, and why cannot the RH positive can donate the blood to RH negative. Fine. So with this we have completed the ABO blood grouping. Now we will be reading about what? The clotting mechanism of blood, which is again a very interesting topic. Okay, what exactly it is? The clotting mechanism of blood, or we can say the blood clotting. The blood clotting. Fine. Now what exactly happens in blood clotting? Yeah, so what exactly is the clotting? So here it stops, you know, the blood, for example, whenever there is an injury, whenever there is an external injury, so at that time what will happen? So at that time the blood, okay, would be oozing out, okay, and to prevent the loss of blood from the body, this clotting mechanism happens. Now how exactly, you know, for example, whenever you may have suffered from some small cut, okay, so it, after some time it will stop automatically. Now what exactly, okay, happens in this process? Let us try to understand in detail. Now before that, tell me one thing, is clotting and clumping both the words same or different? Okay, I am saying clotting and clumping are both same or both different. So let me tell you, both the words are different. Okay, the clumping is when the cells stick together. The clotting is when you prevent prevent the blood from the loss by finding fine mesh-like structures. Okay, so that we will be discussing how exactly this process is being carried out in the body. Fine. Now, for a clotting to happen, what need to happen first? First, the injury. First, there need to be an the injury and the loss of blood. Now here it is followed by the blood clotting. Now here there will be two pathways. Fine. Whenever there is an injury, at the site of injury, okay, there are two pathways being followed. What is the 2 pathway? We have this extrinsic pathway. We have this extrinsic pathway, and then we have what? The intrinsic pathway. Okay, we have this extrinsic pathway, and then we have what? The intrinsic pathway. In this 2, the process will be happening. Now, what exactly happens? For example, this is a site of injury. Okay, this is a site of injury. Yeah, this is the site of injury. Now, the tissue which is present here is completely what? Damaged. Fine. Now, apart from that, the blood vessels which are present inside. Okay, the blood vessels. Now, here if there is a damage. Okay, now here if there is a damage, now what will happen? There is a loss of blood. Okay, right? There is a loss of blood. Now here what will happen? Now this tissue will be releasing what? The tissue thromboplastin. Fine. Whereas here they will be releasing what? The platelet thromboplastin. Platelet thromboplastin. Now this will, both of them, they'll react, and what they'll do? They'll increase. Okay, they will be increasing the number of thrombocytes. Okay, thrombocytes at the site of injury. Okay, at the site of injury. Right. Now what will happen? Now here this thromboplastin, through the cascade of mechanism, will be forming what? Now here the cascade. Okay, the series of steps, the cascade will be forming what? The thrombokinase. What they will be transformed into? They will be transformed into what? The thrombokinase. Okay. Okay. Fine. The thrombokinase. Now what this thrombokinase will be doing? Now here this will be happening in the presence of what? The calcium. Right? Now what this thrombokinase will be doing? So we have this inactive prothrombin. We have this inactive prothrombin. Inactive prothrombin. This will be converted into what? The thrombin. This will be converted into what? The thrombin. So the inactive, the prothrombin in the presence of the thrombokinase will be what? Converting into what? The thrombin. Now what the thrombin will be doing? The thrombin, okay, so here it will be convert. Now this will convert the inactive fibrinogen. Okay, into what? It will be converting the inactive fibrinogen you have this thrombin. Yeah, it will be converting the inactive fibrinogen into the active fibrin. Into what? Into the active fibrin. Okay. Okay, it will convert the inactive fibrinogen into what? the active fibrin. Now what this fibrin will be do? Now this is insoluble. This fibrin is what? Insoluble. What do you mean by insoluble? Means it is not soluble in the body. Now here what will happen? Okay, so here this mesh like structures, these are fibrins. Now these fibrins will be forming a mesh like structure, right? Now where exactly? At the site of injury. So here I can say the site of injury. For example, let us do it up here. Yeah. This is a vessel where exactly there is a cut. So these insoluble fibrins, they will be forming a mesh-like structure. Okay, they will be forming a mesh-like structure. Now what will be trapped inside this mesh? This mesh inside, they will be trapped on by the platelets. You have the platelets, right? Then you have some WBCs, you have some RBCs. Okay, so all these are trapped and forms what? Forms a mesh-like structure. This is what? The mesh. Now it will be again converting to what? The hard scum. Now sometimes whenever there is an injury and when it dries up, you feel something brown color over it, right? Now this is the brown colored, fine, that is called the scum there. So the brown color would be formed, right? That is called what? The scum. Now it will be helping in what? The healing there. So this is the mechanism of the blood clotting. So let us quickly revise it out. We have what? At the site of injury. So the two pathways are being formed, the extrinsic and the intrinsic pathway. So at the site of tissue, where at the site of injury, the tissue would be forming, releasing what? Tissue thromboplastin, and the blood vessel will be releasing what? The platelet thromboplastin. Now both of them, they will be sending a signal to the thrombocytes to accumulate at the site of injury. So at that time, what will happen through the cascade of steps, they will be converting into what? The thrombokinase. Now in the presence of thrombokinase, what will happen? In the presence of thrombokinase, this inactive prothrombin. This inactive prothrombin will be what? Converted into the thrombin. Now only in the presence of thrombin, what happens? We have this protein. You remember when we were discussing about this proteins which are present. Okay, so what we have? We have the albumin, we have the globulins, and then we have the fibrinogen. Now this fibrinogen is converted into what? The fibrin, right? Now these fibrins, they are insoluble, means they are not soluble in the body, they form a mesh-like structure. Okay, here you can see now what is trapped inside the mesh-like structures, the WBCs, the platelets, okay, the dead RBCs, and here what will happen? They'll form a mesh-like structure fully covered, and here hence they prevent the the flow of the blood, right, the loss of the blood. Now here what will happen to this mesh? It will dry up, and once the wound heals, it forms a hard outer structure called the scum, which is brown in color, and that scum over a period of time, it will fall off by itself, and here at the site of injury you can see the development of the new skin there. Okay, so this is the mechanism, the blood clotting mechanism, right? So here please student, the clotting only happens when there is an injury, and the clumping and clotting both are different words. Please don't get confused by it. Okay, now next let us move on to the next part, which is what? The lymph. What exactly is the lymph? What exactly is a lymph? What exactly is a lymph? Have you heard of lymph? Right? So now we will be reading about the lymph. Now what exactly is the lymph, students? Lymph is also a fluid connective tissue. Okay, it's a specialized. Now here we have, once you read about this tissues part, we have 4 different type of tissues. We have epithelial, we have connective, we have muscular, we have neural. In the connective, we have the loose connective tissue, dense connective tissue, and specialized connective tissue. In specialized, we have skeletal connective tissue and fluid connective tissue. So in fluid connective tissue, we have this blood and the lymph. In the skeletal, you have bone and the cartilage. Okay, so in the fluid connective tissue, what you have in the fluid connective tissue, you have the blood along with the lymph. So here you can say that, okay, so what exactly is the lymph? You can also say it's a fluid connective tissue. Now, where exactly, what is the color? Is there any color? Okay, now where exactly can you spot the lymph in our body? Now think of it. Yeah, so like for example, I'll give you one good example, so try to connect to it. Now for example, you know, now when you are taking the online class, you have been sitting in front of your laptop screens, okay, or your desktop screens, or also your mobile phones. Now you are prolonged sitting for 3 to 4 hours without making any movement. So at the time what happens, you feel some numbness in your legs, or for example when you sit on the floor crossing your legs. Okay, when you sit onto the ground floor, I mean on the floor, crossing your legs for a 20 to 30 minutes, what would happen? You start feeling this numbness in your feet. Okay, and also a tingling sensation, right? So for example, when you start walking, soon after you sit for 20 minutes on the floor, there is some tingling sensation there, and also there is what, some numbness. Now why exactly, have you ever wondered it happens? Yeah, so let me tell you, it happens because of the accumulation of a fluid. What is the fluid called? The interstitial fluid. Okay, and the lymph there. It happens due to the accumulation of the lymph there. Now here we have a total system of the lymph. We have the lymph, which is a fluid, then we have a lymph vessels, then we have the lymph, you know, the lymph capillaries which open to lymph vessels, and we have the lymph duct, and we have a total lymphatic system. Okay, but now you will be only reading about the lymph as it is a CBC class. So in the 11th, when we are teaching the chapter body fluids and circulation, we'll be explaining about you in detail. So here you can write down, it is a colorless fluid. It is a colorless fluid, means what exactly is the composition? So you can write, the lymph consists of, it consists of the plasma. Okay, you can write down the plasma. Next you have the WBCs, right? And then you have some nutrients. Next you have some hormones, right? And the oxygen. And what? The oxygen. Ma'am, how exactly is this lymph formed in our body? So let us try to understand this. Now students, please have a look here. Now here we have an artery. Okay, now here this is an artery, and what we have this tissue. Okay, what is this? This is a tissue, and this is your, this is your capillary. This is your capillary which is carrying the blood inside it, right? Which is carrying blood inside it. Okay, you have the blood which is carried in this way. So what will happen due to the pressure? Okay, so because the heart pumps the blood through the pressure, so here due to the pressure, now see what will happen. So now from this they need to enter into what? They need to enter into the veins, right? Yeah, they need to enter into the veins. So here what happens due to the pressure? So due to the excess pressure, or you can say high pressure, some fluid, okay, will be coming out of this capillary and getting around this tissue. Okay, getting accumulated around this tissue. Now this is called what? Your interstitial fluid. Interstitial fluid. Okay, now the interstitial fluid will be rich in the nutrients. Okay, it will be rich in the oxygen, it will be supplying, because the one capillary cannot go through the many cells which are present inside, right? They are just peripheral. Now if they need to transport deep, okay, there is multicellular, we have many layers also, so we need this interstitial tissue. Now what will happen? Here we have a capillary. What is a capillary? This is a lymph capillary. What is this capillary? The lymph capillary. Okay, so these interstitial fluid, what will happen? Okay, after some time it starts entering into the lymph capillary. Now once this interstitial fluid enters into the lymph capillary, at that time it is called what? At that time it is called the lymph. Getting my point? Yeah. So first we are getting interstitial fluid, then when it enters into the lymph vessel, it is called the lymph there. Fine. Now what happens is lymph vessels open into lymph capillary opens into the lymph vessels and goes into the lymph duct, lymphatic ducts, and it drains out into what? Into the vena cava, and again get mixed with the blood. Fine. Getting my point? Now here if I say the component, the plasma component of the lymph and the mineral constitute, the mineral composition of the lymph and the plasma is one and the same. Okay, so the minerals which are present in the plasma and the minerals which are present in what? Which are present here in the lymph is the same. The mineral composition is same in both of them. And you remember in the grade 10th you had a chapter called life process. In the life process when you were discussing the digestion topic, so at that time you have studied now food get digested in the stomach, it is getting absorbed in the small intestine. Now what structures help in absorption? That is the villi. Okay, now in the villi you can see the vessels, the lymphatic vessels called what? The lacteals. What they are called? They are called the lacteals. Now lacteals, what exactly they do? They absorb the fat from the body. Yes or not? What they do? They absorb the fat from the body. So here what they will be doing? The lymph is actually important because for the transport of what? The oxygen, nutrients to what? All the tissues there. Okay, so again here when it enters into what? The lymph vessel, it is called the lymph, lymph capillary, it is called the lymph there. Okay, now let us quickly go through the the flow chart, how exactly it does. Now what I am telling you, now this is a blood vessel, right? So you have the blood which is flowing in. Now here due to the pressure what will happen? Some of the liquid part flows out. Now this is called the interstitial fluid. Now this fluid is called what? This fluid is called the interstitial fluid. The interstitial fluid. Now this interstitial fluid when it enters, what happens when it enters? When it enters into lymph capillary, it is called what? It is called lymph. Very important. Fine. Okay, so here this interstitial fluid, it enters into the lymph capillary, next goes into the lymph vessel, next open into what? The lymphatic nodes, next the lymphatic duct. This lymphatic duct open into what? Open into this vena cava. Okay, I'll write it down here. Yeah, I'll write down here. So it opens into what? The inferior, the superior and inferior vena cava. Inferior vena cava. And what happened? Opens into right atrium and gets mixed with your deoxygenated blood. Deoxygenated blood. Clear? Fine? Okay, so here interstitial fluid, lymph vessels, lymphatic nodes. You know lymphatic nodes. What are lymphatic nodes? Okay, so for example, you can say your tonsils, right? The Peyer's patches. Then you have the spleen. Yeah. What are lymphatic organs? Any idea what are lymphatic organs? Lymphatic organ. We have this thymus. Right? Okay. Then we have what? Okay, the thymus is primary. Then we also have this, the bone marrow. Right? So they are primary lymphoid organs. Right? So this is the one about the lymph. So I think this is clear, right? What is interstitial fluid? Okay, it flows through the lymph capillaries, right? through this lymph vessels, lymphatic nodes and the lymphatic duct. Got my point? So I hope this is clear. I hope this topic is clear for all of you. So we have completed the ABO blood grouping and the blood clotting mechanism and also the lymph. In tomorrow's class, students will be studying about the human circulatory system, the structure of the heart, now how exactly the blood is pumped. Okay, so that's all for today, students. Thank you. We'll again continue tomorrow.